Hi guys, welcome to this week's discussion. We're going to be talking about photosynthesis, particularly the light reactions as well as the Calvin cycle that makes up the main components of photosynthesis. So the first thing that I wanted to go over is the key objectives of what we're trying to take away from this discussion section today. So I want you guys to be able to be familiar with the inputs and outputs of each of the cycles, the path of the electrons, where the electron's flowing, how that relates to free energy especially will be really important. Where each process occurs in the cell. I don't want it to become so far, so in depth that you guys forget like the concept of where we actually are physically. I want to go over what antenna pigments are and reaction centers and how those make up a photosystem. How resonance occurs and how it relates to the principles of free energy. I want to talk about cyclic versus the non-cyclic photophosphorylation and why that happens. I want to go over the three phases of the Calvin cycle, what photorespiration is and why it occurs and why it's bad. And I want to just generally get you guys to think about comparing and contrasting to mitochondria, because chloroplasts and mitochondria function pretty similarly. Okay, so first I want to just show the picture of a chloroplast. So here is our chloroplast. We have this outer and inner membrane, as you can see. We have these thylakoids here that kind of stack up into a little stack called the granum. And inside the thylakoids, we have the lumen. And outside the thylakoids, we have the stroma, which is an aqueous fluid that's kind of synonymous with the cytoplasm of a cell, but it's inside the chloroplast. Okay, and one other thing I wanted to talk about in terms of location is photosystem 2 and photosystem 1. So remember that the light reactions start actually in photosystem 2 and then go to photosystem 1. So it's a little bit reverse about what you would think. So just try to remember that. And so think about how the actual light reaction occurs. So we know that the photosystem 2 happens first, and it creates these intermediates that are then shuttled into photosystem 1. The overall products of the photosystem 1 are then shuttled out to the Calvin cycle. So if we remember, the light reactions take place inside the thylakoid membranes. So all up on these little membranes in this entire stack of granum. The Calvin cycle, on the other hand, takes place in the stroma itself. So structure being synonymous with function a lot of the time, the photosystem 2, the first one, it's going to be kind of located in the middle here. It doesn't really need to have that much contact with the stroma because it's not really directly interacting with the Calvin cycle or shuttling products to it. So it doesn't really matter if it's located kind of in the middle here without much access to that. The photosystem 1s, however, are shuttling their products directly into the Calvin cycle, so they need to be able to push them into the stroma. So they're going to be located more on the ends here. And so we can see that in this picture where we see the orange is the photosystem 2, and the reds are the photosystem 1. So you can kind of see that these are located more on the outsides versus the middle. Okay, so going over chlorophylls and what they are, they're essentially just a type of colored compounds that are available in chloroplasts that capture light energy, which allows photosynthesis to occur. So generally, we're talking about chlorophylls, but there's also other things like carotenoids and xanthophylls and several other different types of accessory pigments, but mainly it's chlorophylls. And so here is a picture of the absorption spectra of different types of pigment molecules. And we can see that there's a lot of peaks over here in this area, in this green, almost greenish blue area, as well as smaller peaks, but still peaks in the reds and oranges and almost veering into the yellows a little bit. And so one important thing to know is that in the fall, in the autumn, when chlorophyll begins to break down because of the temperature and because the length of the days are getting shorter, you're going to see a lot of these chlorophylls are starting to break down. And when that happens, the colors from the accessory pigments are going to actually show up more. So that's why we see a transfer from this area of the visible light spectrum over to this. And the important thing to remember is that the fact that plants use light only from the visible spectra is kind of just having to do with the wavelengths and the energy of the wavelengths of light. So infrared light is not going to provide enough energy for the photosynthesis reaction to occur. And ultraviolet light is going to be so energetic that it's going to actually it's going to actually spur creation of free radicals, which is going to be really harmful to the cell. So plants actually have defenses against UV light because it's so damaging. So that's why we're kind of here in this absorption spectra. And on the next slide, I'm going to go over kind of why we have these clusters of peaks in these two different areas. Okay, so here is a little chart that shows exactly how photons are able to provide energy to the pigment molecules. So you can see here, we have this pigment molecule in its ground state. It's kind of just chilling, nothing's happening to it. And then it gets excited by a photon of a certain wavelength. And it's able to be energized into an excited state. And what this really means is that one of the electrons of the molecule is being pushed out to a higher energy orbital. And so the reason why we have those two concentrated peaks is because this is the energy required to bump up from one orbital to another in some of the molecules. And then this is from one orbital to another in the other molecules. So like we have these like specific jumps that are required to get from one orbital to the next, which is why we don't see a similar like level of peaks in the middle here. This is like one orbital to another, and this is one orbital to another. Okay, so what a photosystem is essentially the conglomeration of the antenna chlorophyll, the reaction center, in the thylakoid membrane. So we can see here, this is where the antenna chlorophylls are. This is where the photons are going to be striking these pigment molecules and causing them to become excited. Now, the actual energy that's coming from this isn't actually due to this contact with the pigment molecule itself, like by the photon. It's actually coming from when this excited electron in this high energy orbital, when it falls back down to its natural state. So the energy is being provided when it goes from an excited, agitated state to a lower agitation state. When that happens, we can think of the principles of free energy. When you're going from this high excitement and agitation and low stability to a sense of higher stability, then that's going to be exergonic and release energy. And so that energy gets released and touches its neighboring chlorophylls, which then causes a similar reaction where they have their electrons being pushed out to a higher energy orbital, and then when they return to their ground state, they like push it on further. So it's kind of like a domino chain reaction. And so we can see that here. This is an example of like, okay, this one is touching this one, which is touching this one, and blah, blah, blah. Now, the reason why the photon can't just directly strike the reaction center and cause the reaction to occur is because we need this buildup of energy in order to power a reaction that's going to be strong enough to generate ATP. So we can think about what we talked about in cell respiration, where a similar type of buildup of energy was occurring when we had that proton gradient. We had an electrochemical gradient across the membrane, and that was kind of a reservoir that provided all that energy that allowed us to generate ATP. Same thing's happening here, except the energy is coming about differently. It's coming from these photons. Okay, so this is another example of what resonance looks like. So resonance is just that whole process of energy transfer from these different pigment molecules. And you can see that each one of these steps is generating energy, which is why we like having these really long chains rather than just, oh, a photon is hitting the reaction center. So this is a figure of the Z scheme. And this kind of gives a broader view, I think, of how this energy is powering photosynthesis altogether. So we can see, okay, here we start with water and light as an input. The light in the form of photons, we know, is undergoing resonance. And this water here is being oxidized. So it's losing electrons because the electrons are being donated to the photosystem 2, which is the P680. So we have this resonance occurring as well as this electron donation to the photosystem. This process here is going to be a main cause of oxygen, which is really important for us, right? For humans and for other like animals and things that can't photosynthesize, we need oxygen. And then, so now we have these really highly energetic electrons here. And what that's going to do is it's going to donate these electrons to the pheophyton in order to return to a stable state. And so now this pheophyton is super energized. So we see that we're at this really high level of energy. This is a gradient of electron energy here. And then we're going to use the electrons here, and we're going to fall down an electron transport chain, which is going to provide the power to generate ATP, which is really cool. So we have this pheophyton. The electrons are going to be transferred to peak Q, which is plastic quinone. And the reason why it's able to do this without any sense of, or like without any input, energy input, is because this is going to be more electronegative. So the electrons are automatically going to go here without any sort of extra input required. So we're getting to a more stable state. And then the electrons are going to go into the cytochrome complex, B6F. And at this stage, something really, really important is happening. And I'll actually move ahead to the slide here. You can see that from this PQ to the cytochrome, what's happening is that that energy that's being released here is being used to power the movement of protons against its gradient into the lumen. So from this transfer, we're able to push this proton here, which is in the state of almost no other protons. And then here we have this really high concentration of protons. So it's a concentration gradient as well as a charge gradient, because you have all of these positive charges. But since we have this flow of energy, we're able to use that to power here. Now this is really important again, because storing all this energy up is like creating a reservoir that we'll be able to use to generate power. And we can see that this is going to be really similar to what we saw in oxidative phosphorylation in mitochondria, because it's actually the same enzyme here, this ATP synthase, that when these extra protons are being pumped through in the flow of its gradient, because it wants to go from this side to this side in order to be neutral. When it does this, this provides so much energy that we're able to form ATP here. So that's why this part is really important. And so now, after we had the cytochrome that's pushing in these hydrogens, we transfer the electrons to PC, which is plastocyanin. And the really important thing about plastocyanin is that we can see by its structure that it's not actually attached to this membrane. And this figure is kind of an exaggeration in terms of how close these photosystems are to each other. And we can see that the photosystems itself are attached to the membrane, so they don't really have motility. So this plastocyanin, its function is to shuttle the electrons that are from this process into this process. So it's super, super important, because if we didn't have this, we wouldn't be able to have this occur, because they would just be kind of stuck over here in this area. Cool, so then once we have the plastocyanin shuttling over to the next photosystem, photosystem 1, remember that, it's photosystem 1 here. We're gonna donate the electrons, again, to photosystem 1. Light is going to be input, and same sort of phenomenon with the resonance happening. So all that energy is going to be passing along the pigment molecules, like here, like this. And then remember that this is going to be super energizing to have all of this light energy here. So the electrons are going to be highly energized, and they're going to fall down an electron transport chain again. So they're going to be passed to ferredoxin. Under normal conditions, ferredoxin is going to donate its electrons to NADP reductase, which is an enzyme that will take NADP that we had in the cell and transfer it into NADPH. So that's what generally happens. But there's also an alternative path that can happen, which is called cyclic phosphorylation. Now, cyclic phosphorylation is sort of a feedback loop. And it's used to prevent photosystem 1 from producing too much NADPH. So we can see here, if this was all that happened, the only way, or like the only path that electrons could take once they reach photosystem 1 is to end up as NADPH. The problem with that is the Calvin cycle can be overloaded, whether it doesn't have enough ATP or it's being so highly energized by like a bunch of light that it can't keep up. If we have a buildup of NADPH here, these can start reacting to form free radicals or reactive oxygen species. And those can be really damaging to a cell. It's actually the same sort of damage that would happen with ultraviolet light. And we remember that these cells have UV protection kind of built in because they don't want those reactive oxygen species to occur. So this would be really damaging if we didn't have a regulatory mechanism to combat that. But luckily, we do. So the ferredoxin will be able to detect the ratio between this NADP and the NADPH in the cell. You don't really have to know the mechanism, like how that happens. If you're interested, let me know. But it's a little bit beyond the scope of this course about how it actually does that. But if it recognizes that NADPH is outweighing NADP, it's gonna kind of be on high alert because we know that we don't want that. And what it'll do instead is it'll take the electrons from the photosystem 1, and instead of shuttling them to this reductase, it's going to shuttle them back towards the plastiquinone here, which if we remember from the Z scheme, we're basically taking from here to here, which is at a relatively high place in the electron transport chain. So then the electrons are able to be fall down this chain again and produce ATP. This is really nice on a couple of accounts because one, you're protecting yourself from reactive oxygen species, but two, you're also producing more ATP in the process, and we know that ATP helps power the Calvin cycle. So if you had too much NADPH but not enough ATP to conduct the Calvin cycle, you're kind of it's almost like a negative feedback loop. You're stopping the NADPH production, but you're increasing the amount of ATP, which will hopefully spur the Calvin cycle back on again, and then you can resume your non-cyclic photophosphorylation. Cool. So overall, we want to think about, okay, in the light reactions, what did we put in and what did we get out? So if you remember, we put in those photons, that initial photons that will hit the photosystem 2, and water, which was oxidized, remember, because it was losing electrons to the photosystem. And what did we get out? When we did the non-cyclic photophosphorylation, we got out oxygen from the first step, but we also got the ATP from the photosystem 2 electron transport chain, and then the NADPH from the photosystem 1 electron transport chain. So we can look back here. You can see, okay, when we had this input, when we donated the electrons, we got oxygen here. We got ATP from this first electron transport chain, and then we got the NADPH from the second electron transport chain. So those are our overall outputs. If we were doing cyclic photophosphorylation, the only output that we would have is ATP. And this is because we would be just doing this step here. So we wouldn't be getting any of this oxygen formation or this NADPH formation. But remember that this is just under a specific circumstance. You're not always going to be doing cyclic photophosphorylation. Okay, so moving on to the Calvin cycle. The Calvin cycle is the next step of photosynthesis. So essentially all the products that were formed from the light reactions are going to be inputs that are required for the Calvin cycle to start. So Calvin cycle occurs in the stroma, remember, so that's why the location of the photosystem 1 is so helpful, is because it'll be directly able to shuttle their ATP and the NADPH and stuff to power this cycle. It consists of 3 different phases: the carbon fixation phase, the reduction phase, and the regeneration phase. So in the first phase, which is carbon fixation, RUBP, which is our starting molecule, interacts with the CO2 that's being input from the environment. So CO2 is one of our inputs. And it's a reaction that's catalyzed by Rubisco to form 3-PGA, which is 3-phosphoglycerate. And so we can see this here. You have the CO2 being input, Rubisco is the enzyme, and then you're being fixed into this 3-phosphoglycerate form. The next step is reduction. So we have this 3-phosphoglycerate to start, and then we are reducing it into G3P, or triose phosphate, but it's going to be an endergonic reaction. So we need to power it with a couple sources of energy, with ATP in this first step and with NADPH in the second step. So we can see that the reduction phase is going to have this intermediate biphosphoglycerate. Okay, so after 6 turns of the Calvin cycle, you're going to form 12 G3P molecules total. 2 of them are going to leave the cycle, and they're going to enter the cytoplasm, and they're going to interact with each other to form glucose. And so that's what we want. This is like our ultimate carbohydrate formation. And then the other 10 are going to go into phase 3, are going to be funneled into phase 3. Phase 3 is the regeneration of the electron acceptor, which we remember was our first initial molecule, the RUBP, which is necessary in order for the cycle to continue. So we want that to be regenerated, so we'll always have a source of being able to drive the cycle forward. This also requires a little bit of ATP, 3 ATP, so it's less energetically expensive than the phase 2, but it does still require some. And so this will require 10 G3Ps, which you remember there was 12 total, 2 left to form glucose, and the rest of the 10 will go through this phase and will ultimately regenerate 6 molecules of the RUBP for every molecule of glucose that was formed. So we can kind of sum this up in this concise little slide. Formation of 1 glucose is going to require how many turns of the Calvin cycle? I believe we said 6. So our input is 6 CO2 molecules, 18 ATP, and we can see that if we look at phase 2. So we're going to have 6 ATP here, and then it's going to be 2 cycles, so 12 here, plus 6 from here, because we have 3 ATP required for each G3P molecule, and we need 2 G3P molecules. So 12 NADPH, and then our outputs are ultimately going to be this 1 glucose by way of having those 2 G3P molecules that are exiting the cell, 18 ADP, 12 NADP, and 6 of the original electron acceptors, the RUBP. So that's kind of our summation slide. An important thing to note is that photorespiration can also occur in the Calvin cycle. It's not ideal, but Rubisco, the enzyme that we remember from phase 1, is able to accept either oxygen or carbon dioxide as a substrate. So we remember when we were talking about the Calvin cycle, we want it to accept carbon dioxide, its input, so that it can form our product of interest, but it can also accept oxygen. So in this case, oxygen is acting as a competitive inhibitor because it's able to bind to the active site of the same substrate as CO2. And it's an inhibitor because it's inhibiting the amount of the reaction that we actually want, which is the CO2. So the problem with photorespiration, the reason why we don't like it, can be seen here. So if we have this RUBP and we perform the reaction that we want, which is CO2, we're getting these 2 PGA molecules, which can then be further used in the Calvin cycle, and we'll get the exact same outputs and stuff that we previously described. However, if we get photorespiration, where we get this oxygen being reacted with Rubisco instead, we only get 1 PGA. And then we get this one other molecule that is essentially a waste product. It can't be used in the Calvin cycle. So it's just generated and lost. So not only do we have this formation of something that's kind of wasteful, but we also will have to undergo more turns of the Calvin cycle because we need 2 PGA, and here we just have 1. So it's going to be way less efficient because we'll need to turn the Calvin cycle more, which will require more ATP and NADPH in order to get to the same number of PGAs. And we can see here, this is a good picture of it. So we have this oxygenation step instead of carbon fixation. And then this is going to be our waste product. And then this will continue on as expected, but you're going to get fewer carbohydrates in the form that we want, in the triosphosphate form. So that's why we don't appreciate Rubisco as much, because it can undergo these 2 reactions, one of which will inhibit the other and is not favorable. Cool, so that's basically all that I wanted to cover in terms of concepts. So hopefully this made a little bit of sense. And then you can go through the discussion exercise and quiz and turn those in by the end of the day. And let me know if you have any questions. So thanks for listening, guys.